All right, so this is our biomolecules review. Your nucleic acids go to nucleotides. Your lipids go to fatty acids and glycerol. Your proteins go to amino acids. And finally, your carbohydrates go to monosaccharides. A nucleic acid is made up of a monomers called nucleotides. The key word here is nucleic acid. Monomers of nucleic acids are nucleotides. Examples of nucleic acids are DNA and RNA. Keyword here is nucleic acids. We're also going to add ATP because I didn't really mention it in the slides, but ATP is a nucleic acid. There are 20 different types of amino acids. That can be found in proteins. The keywords here are 20 because there are 20 amino acids, and protein. Amino acids make up protein. A carbon-hydrogen chain that makes up part of a lipid is called a fatty acid. The keyword here is lipid and chain. It's a fatty acid chain. An enzyme, which is a molecule that speeds up reaction, is an example of a protein. And the keyword here is enzyme. Enzymes are proteins. Glycogen, which is an energy source. Energy is the key word. It's an example of a carbohydrate. Again, the key word is energy. Carbohydrates give us energy. A small building block of a macromolecule is called, keyword is small, that's a monomer. Which joins together to form a large molecule. Large is the key word, a polymer. All right, now we're going to go on. Which macromolecule, Which macromolecule does not contain nitrogen? Remember in the video, they talked about CHO, CHON, and CHOMP. CHO was for carbon, the second one was for lipids, the third one was for proteins, and the last one was for nucleic acids. Proteins contain contain nitrogen. Nucleic acids contain nitrogen. The only one that does not is your carbohydrate. Which macromolecule does not provide energy? Remember, carbs is your quick energy. Lipids is your long-term energy. The only one that is not energy is your nucleic acids. Which is not an example of a lipid? Triglycerides are long fatty acid chains. Phospholipids has the word lipid in it. Remember, that's with the head and the tails that make up the membrane. Disaccharides are sugars, and sugars are carbs. So a disaccharide is not a lipid. What is not a polysaccharide? Well, a phospholipid, again, it has the lipid. Cellulose, anything ending in OSE is a sugar. And a starch is a carb. And a polysaccharide, that's also a carb. It's a sugar. So the only one that's not a carb, that's not a sugar, is our lipid. What is this an example of? We have a hexagon and a pentagon. Let's talk about what these words mean. Monosaccharide, mono means one. Disaccharide, di means two. Polysaccharide, poly means many. So when we're looking at this example, we see that we have two different carbohydrate links. So it's a disaccharide. Two carbs. All right, now we're going to sort these into their correct columns. We have carbohydrates, lipids, proteins, and nucleic acids. I'm just going to write a C for carb, an L for lipid, a P for protein, and an N for nucleic acids. So my carbohydrates. In your column, you should have put made up of monosaccharides. Monosaccharides are carbs. Main energy source. Energy tells you that's a carb. Down here, commonly called sugars. Sugars are carbs. And over here on my top right, lactose and cellulose ending in OSE, that's a sugar, which is a carb. For my lipids, oils and fats, those are lipids. My fatty acids and glycerol, those are also lipids. Triglycerides are lipids. They make up the cell membrane with their phospholipid bilayer, so that's my lipid. For proteins, blood, skin, hair, and muscles, that's protein. Makes enzymes, enzymes are protein. Contains a peptide bond, that's also protein. And made up of amino acids. Remember, there are 20 different amino acids for protein. And lastly, we have nucleic acids. Remember, ATP is a nucleic acid. Nucleotides are monomers of nucleic acids. DNA and RNA are nucleic acids. Contains a phosphate, that is also nucleic acids. Remember CHO, CHON, and CHOMP. The only one with a P is my nucleic acid. All right, so now we're going to match these molecules. The first one has a phosphate group, a sugar, and a nitrogen base. That's our nucleotide. The second one has an amino group, a R side chain, and a carboxyl group. That is going to be our amino acid. And remember, there's 20 different kinds of these. Next, we have a long chain. We actually have 3 long chains. So that's going to be our triglyceride. Tri means 3. And then we have our sugar. Sugar is a monosaccharide. It's just one sugar. Now we're going to fill in our blocks for our 4 major biomolecules. The monomers of carbohydrates are called monosaccharides. An example of that would be glucose. Everything ending in OSC is a sugar. And it's for our quick, short-term energy. It also functions for structural support in plants with cellulose and for food storage. Our lipids, the monomers include glycerol and fatty acids. Examples would be fats, steroids, and phospholipids. And this is for long-term energy. Lipids are also important for the cell membrane and for insulation for the body. Proteins. Monomers include amino acids. Remember, there are 20. Examples include hemoglobin, insulin, and thyroxine. Proteins are important for a catalyst. They make enzymes, hormones, and muscle movement. Finally, nucleic acids. The monomers of nucleic acids are called nucleotides. Examples are going to be your DNA, your RNA, oops, and it's also going to be ATP. Don't forget that. It stores your genetic information. It's also responsible for gene expression. All right, next we're going to start with our body systems. Hopefully, again, these are easy to tell which one's which. Your digestive system is going to include your mouth, your stomach, esophagus, and intestines. Your circulatory system is going to have your heart, your veins, and your artery. And your endocrine system is going to have all of your glands. Next, we're going to put the organs to their correct body systems. For digestive, I'm going to use D. For circulatory, I'm going to use a C. And for the endocrine, I'm going to use an E. Digestive system is going to include your large intestine, your mouth, your stomach, your liver, your gallbladder, rectum, teeth, and esophagus. Your circulatory system is going to have your blood, veins, arteries, ventricles, your aorta, and your heart. Finally, your endocrine system is going to have all of your glands, your pineal gland, ovaries, uterus, adrenal gland, the pancreas, thyroid, and your testes. Next, we're going to match the systems with their correct definitions. Your digestive system has organs working together to convert food into energy and basic nutrients to feed the entire body. Your endocrine system is going to use glands that produce and secrete hormones that the body uses. And finally, your circulatory system is going to allow blood to circulate and transport nutrients to the parts of the body. Next, we have an example of enzymes helping with activation energy. These levels tell us the amount of activation energy needed. This top one needs a lot of energy. And this is an activation without an enzyme. It needs more energy because it does not have an enzyme to help it out. The lower one needs less energy, and that's because it has the help with an enzyme. It has an enzyme, so it does not need as much energy to get the products created. Finally, after the reaction's done, we're going to release our energy. Our energy released. And lastly, we're going to talk about enzymes. Our enzyme is going to function like our lock. And our lock is going to bind to a substrate, which is our key. This is later going to turn into products after it's done with the enzyme. And our substrate and our enzyme are going to bind together at the active site. So we've talked about inhibitors that block enzymes. Now we're just going to have specific names for them. The first one is a competitive inhibitor. And this binds to the active site, so it blocks the active site. It doesn't let anything else come into the active site. And the second one is a non-competitive inhibitor. This one doesn't block the active site, but it does bind to a different site on the enzyme. It attaches it somewhere else, and it causes the active site to change. So that way the lock and key do not match up together anymore. And finally, enzyme activity can be affected by your pH, your temperature, and I also talked about the chemical levels, which tell you the concentration of substances. All right, so that's all I wanted to review before your test tomorrow. If you need help studying, make flashcards. You can go online and make an online Quizlet at Quizlet.com. And you can also read your review over and over again until you feel comfortable and until you understand everything. I will not be here tomorrow, Friday, the day of your test. I will not be able to grade your tests until Tuesday. So technically, you could turn in your test as late as Monday, and I will not notice because I will not see them until Tuesday when I come back. So if you want to take your time and have extra days studying, be my guest. But make sure you get the test done by Monday.